Correct.
Great.
Okay. All right. Good morning, everybody. I'm going to today continue with the bioremediation lecture. Last week, we talked about, biodegradation processes and bioremediation. Today, I'm going to talk about some case studies. I will review a little bit about basic bioremediation, and then we're going to talk about few case studies. And then the last two case studies I personally involved in my research. I'm going to discuss that today, okay? So last week we defined bioremediation as use of biological organisms or their product to enhance environmental cleanup. But specifically what we are most of the time using is subsurface microorganisms, microorganisms in the soil and below soil, to reduce the hazardous nature of chemicals, convert them into harmless byproducts, okay? So you want to make sure you degrade the chemical. Ideally, you want to mineralize the chemical, convert them into inorganic constituent. In some cases, we biotransform because it makes it less harmful than the parent compound. So we also talked about natural attenuation and enhanced bioremediation by augmentation briefly last week. So again, the key point here, you need to have the bioremediation triangle. You need to make sure you bring all these three elements together: pollutants, organisms, and the environment, whether it is soil, water, air. You bring them all together. That's the engineering challenge in real cleanup process. So stages of biodegradation is, first of all, you need to find out whether there is microorganisms in the site that you want to clean up. So you take some sample, take it to the lab, isolate, and look for the organisms, identify the microbial isolate, and working. Technical difficulties, yeah. And then optimization of degradation condition, you need to determine the efficiency of your microbial consortium. And then you can identify the whole metabolic pathway. Then you want to make sure that you're not activating instead of degrading the compound. And if you want to do some enzyme study, you can do enzyme study, and then you can do more detailed pathway. That's more basic aspect of it, just to know what is going on in the site, okay? So natural attenuation is not very commonly used because it's not fast enough, it's not complete enough, not frequently occurring in most of the site cases. So the current trend in cleanup process is to stimulate the organisms inside. So most of the time, it's a biostimulation process, adding electron donor are some limiting factors like nitrogen and phosphorus. In some cases, bioaugmentation is necessary because the organism is not there. Okay. So let's talk about one of the processes now coming up is phytoremediation. Phytoremediation is, again, everybody knows, is use of the plant for accumulation, removal, or conversion of pollutants. So phytoremediation, broadly, we're using plants, but there are a lot of processes going on. You're stabilizing the chemical that is called phytostabilization. You're volatilizing the compound from below ground through trans-evaporation process through the plant. It goes up in the atmosphere, that is phytovolatilization. And then you have phytostimulation. You're providing the rhizospheric soil microorganism interact with the chemical and you stimulate activity. And then phytotransformation, plant enzyme itself can transform some of the chemicals. So that is phytotransformation. And then the last one is the one that commonly used called phytoextraction, where the plant mobilizes the chemical into the plant biomass, and then you harvest the plant, and then you incinerate the plant to get rid of the chemical. So there are so many processes going on in phytoremediation. Just next picture, you can see, let me see. You can see the plant showing all this phytodegradation happening right on the plant, phytovolatilization chemical go through the plant, and then you have stabilization and phytoextraction stimulation going on. So approximately there are 400 plant species that have been classified as hyper accumulators of heavy metals. So most of the time, phytoremediation is used to clean up heavy metals. So it takes up the heavy metals and you can harvest it, and sometimes you convert the heavy metal from soluble form to insoluble form, and you can precipitate it. So a lot of plants have been used. Some of them are sunflower, grasses, hemp, alfalfa, tobacco plant. Then in aquatic system, water hyacinth is commonly used in phytoremediation process. Again, the root exudates of the plants play an important role because it stimulates microbial activity in rhizospheric soil. And so it can activate some combination of phytoremediation and bioremediation together in the root zone of the plant. Okay. And then genetic engineering, people are using nowadays using greenhouse phytoremediation, where they put bacterial gene in plants, and then they grow the plants to get rid of the chemicals. So some of them been very successful. But again, it's very controlled condition. It's not very broadly used. We're using genetic engineering exactly like in agriculture. Now we produce BT plants like BT cotton, same process applied for cleanup. So bioremediation of metal, most of the time we use phytoremediation for metal removal, heavy metal removal, okay? So we can immobilize the metal, we can bioaccumulate the metal using polysaccharides and lipoproteins. We can precipitate the metal by converting the metal from insoluble to soluble form. And then we can solubilize the metal using siderophores and organic acid and various chemicals produced by plant. So contaminants that are amenable to bioremediations, here's a list. Readily degradable compound are the gasoline component, fuel oil, we have ketone, alcohol, aromatic compound, then two aromatic ring structure compound like naphthalene. And somewhat degradable are creosote and coal tars and pentachlorophenol, a lot of phenolic compounds. The compounds that are difficult to degrade are chlorinated solvents like TCE, carbon tetrachloride, some herbicides and pesticides. Very, very recalcitrant compound that's going to persist in the environment for a long time are dioxins and PCBs. So I talked about the plants, microorganisms, they produce the siderophore molecule to solubilize. So here are some of the examples siderophores. Most of them are produced by Pseudomonas, and Pseudomonas stutzieri is commonly used in solubilizing metals using siderophores. And also, the bacteria produce biosurfactants. The biosurfactants are compound that has both features hydrophobic and hydrophilic, which is nonpolar and polar region in the same molecule that we call that antipathic molecule. So biosurfactants are produced by bacteria, cyanobacteria, fungi, and yeast. And some of the classes of biosurfactants are glycolipids, lipoprotein, phospholipid, glycoprotein, polymeric biosurfactants. So these are widely used for compounds that are not soluble, the compounds that are called DNAPO, dense non-aqueous phase liquid like TCE, to make it more soluble and bioavailable for bacterial degradation. I have a problem today. All right. So the role of biosurfactant is, again, to increase the bioavailability of the compound that are hydrophobic, and also nutrient storage of molecules, and save microbial cells from toxic substances. It protects the microbes, and efflux of harmful compound. The harmful compound can be excreted out quickly by the bacteria, so the bacteria will survive in the presence of toxic nature of the chemical. And also another features of biosurfactant is its extracellular and intracellular interaction. It helps in quorum sensing and in biofilms of bacteria. So those are some of the benefits of biosurfactants with the microbes. So, so we take advantage of this biosurfactant because it's a chemical that are pseudo-solubilized by the biosurfactant. So it also reduces the absorption of non-polar pollutant to the surface of soil particles. So basically it increases the bioavailability for degradation process. So again, quickly go through a couple of this broad technique, in situ, ex situ method. In situ is again, without excavation of the site. Ex situ, we dig up the site and engineer the process. You need to make sure that you optimize the condition. Most of the time, it's aeration. Sometimes, mostly oxygen limiting. Then you need to provide right pH, moisture condition, and nitrogen phosphorus limitation. That's nutrient addition. And sometimes you got to add surfactants to make it bioavailable. Okay. So just again to revisit some of the process, Slurry Reactor can be used. This is really fast process, so you can clean up really quickly, but it's going to cost you a lot of money. Biopile is another process. You can put them in big pile and create an optimum condition for microbes. And then you can use land farming, commonly used land farm. If the compound is leachable, you have to put a liner, plastic liner, and then you create this land farming condition for cleanup. If the compound is not leaching, it's going to stay because of the soil condition. You don't have to dig up and make a clay lay, put a leaching collecting system, but you don't have to do that. Just you can go and do the land farming without disturbing too much. We not only use bioremediation for in soil and water, but you can also use for cleaning up the gaseous chemicals using Bioscrubber, bio-trickling filters, and slow sand or carbon filters. So if there is some gaseous form of chemicals in subsurface condition, you can bring that up and put them in scrubber, and bacteria will use those volatile compounds as carbon source. So here's some of the example of Bioscrubber filters is basically using biological reactor. Instead of putting influent as a wastewater, you're putting influent as inlet gas. And that inlet gas could be all those volatile compounds trapped in your soil. So you kind of collect them and put them in this Bioscrubber. Or you can use slow sand carbon filters. The slow sand carbon filters, you can have bacteria form a layer, biofilm layer. When the chemical go through this layer, they basically use them as carbon source. And the layer contains heterogeneous population of variety of organisms, including bacteria, fungi, rotifer, and protozoa. Okay. So let's look at what kind of chemicals mostly in soil and subsurface contaminant. Most of the time it's oil-related compound like BTAX, benzene, toluene, ethyl benzene, xylene. We have pyrene and other polynuclear aromatics. And then we have chlorinated compound like solvents like TCE and carbon tetrachloride. And then a herbicide and pesticide. And then we have explosive chemical also. So here is a picture to show the various sources where they're coming from. It can come from storage tank, underground storage tank, can be leaking from septic tank, infiltration gallery, landfill leachate. And there are various sources that you can get this pollution, industrial source and landfills and burial areas and dump sites. Okay. So the current issues, as I said, with gasoline use is it's widespread contamination. This is one of the major problems in the US. There are more than 1 million sites that need to be cleaned up. And it's a major threat to drinking water resource because it's going into subsurface and contaminate your drinking water source. Some of the components of the fuels are carcinogenic, it cause cancer. And this problem is not anymore, but we still have pollution. Before we added ethanol to gasoline, in US, we added this MTBE, which is called methyl tertiary butyl ether, which is the oxygenated fuel oxygenator to burn the fuel better and have your tailpipe emission will be less pollution. But now we have this problem because of the gasoline with MTB also in the groundwater. But currently we completely stopped MTB. We now use ethanol, 10% ethanol in the gasoline. So that's a typical fuel spill, most of the time from underground storage tank from gas stations, all right? The gas stations are old, more than 50 to 100 years old, and they're leaking into the groundwater. And most of the lighter fragment of the gasoline float, I call LNAPL, light non-aqueous phase liquid. And then it forms this plume and it's going to spread in the groundwater. So that's a typical contaminated site look like. The second compound that is another problem is chlorinated solvent. These are the degreasing agent. Mostly it's TCE, trichloroethylene. This is just the opposite. This is DNAPL because this compound is denser than water. It'll go to the bottom of the water table and sink to the bottom. It has low solubility, so it will be a permanent source of pollution of groundwater. And it's known carcinogen, and most of the time it's not a primary carbon source for bacteria to use it. So here's a typical example of the DNAPL TCE. Most of the time dry cleaners and also the electronic manufacturing factory, because they use TCE to degrease the circuit board, and also aircraft manufacturing and also car factory. Wherever circuit boards are used, they use TCE to degrease those parts. So those end up in the groundwater. And if you have this kind of problem, you have like clay lens along with groundwater, this TCE collects in the clay lens, and then it's going to drop into the bottom of the water table and become, form an immiscible layer. And then it slowly leaks into the groundwater. It becomes a very permanent source of pollution forever. And so this is one of the major problem in US for cleaning up TCE contamination. So again, the challenge for TCE is the DNAPL is trapped on these clay lenses, and it's hard to get rid of, and it creates a soluble plume for years. Okay, that's a major problem with TCE contamination. So how do you clean up this kind of contamination? You do soil extraction, you do pump and treat. You can do physical or reactive barriers to stop the plume from spreading. You can do air and hydrogen sparging, and then you can use microorganisms, simulate the production of good bacteria. And then, of course, we can, if you have a lot of money, you can use surfactants. So surfactants will make this chemical more bioavailable. So bioremediation is the best way to do this TCE and oil cleanup in subsurface. Because of these reasons, there is no additional disposal cost, low maintenance, and does not create any eyesore, and capable of impacting large area and decreasing site cleanup. So those are some of the advantage of using bioremediation for cleanup of TCE, as well as oil spills in subsurface condition, especially groundwater. Let's talk about a little bit about fundamentals of biodegradation one more time. Just there is no superbug. We cannot use them. We cannot use genetically modified organisms in cleanup. All compounds eventually will biodegrade, but it takes time for bacteria to evolve enzyme. It also needs specific conditions, such as the nutrient availability and whatever is lacking inside. Again, important part is the contaminant should be bioavailable. If it is not bioavailable, the bacteria are not going to go, degrade this compound. And then you need to look for the limiting factors, mostly oxygen, nitrogen, phosphorus. Those are some of the limiting factor in the contaminated site. Then we look for what is happening once you provide all that. The organism is going to metabolize, and you need to look for the metabolic activity of microbes. And then you can choose between aerobic and anaerobic degradation. If anaerobic works for you, just simulate anaerobic conditions and enhance the microbes. And you can reduce the aqueous concentration of contaminant, and then you can reduce the contaminant mass. And these are some of the advantages of using microorganism. The most significant process is reduction of contaminant mass in the system. So you permanently remove the chemical, so you don't have a future liability. So the process is simple. The organism using the contaminant and mineralizing it into carbon dioxide, water, and other salt. Sometimes the biotransformation happens. Example, TCE converted to DCE. Okay. So biodegradation involves a lot of extraction of energy from organic molecules. The bacteria get energy, so they oxidize the compound and they grow on it and make more biomass. Okay? So that's why this is very advantageous using microorganisms for cleanup of these two classes of chemicals. Revisiting the degradation process, there are three ways to do it. The best way is microorganism to use the contaminant as primary carbon source, okay? If that is the case, then that's the easiest way to go, because the organism is using the contaminants such as TCA as sole carbon source, oil component as sole carbon source. Sometimes they use this component as secondary substrate, so they live on other carbon source like glucose, other easily available carbon source, but they also use this as a secondary substrate. Okay, and also the compound is not in high enough concentration in the contaminated site, so they depend on other carbon source. And then the third process is co-metabolic condition. So in this case, you have to provide additional carbon source such as glucose to enhance the bacterial biomass. Okay. So let's look at the requirements for microbial growth. You need to have electron acceptor. The electron donor could be the contaminant itself, or a carbon source could be contaminant itself. You got to have these conditions for bacteria to be happy, right pH, temperature, and then the limiting factor. And sometimes you have to put some trace element. This trace element, most of the time, subsurface is okay. Mostly the limiting factor is nitrogen, phosphorus, and if it is aerobic, the oxygen, okay? So you got to have various electron acceptor option, not only aerobic, you can use the, like last class I talked about nitrate reducing, sulfur reducing, ion reducing condition if there is no oxygen available. And then the pollutant itself could be used as electron donor and the energy source. The bacteria use them and convert them to CO2 and water, and then they use this to grow as well to make more bacterial cells. So again, you can use both aerobic and anaerobic. If oxygen is terminal electron acceptor and the process is aerobic, other processes are anaerobic, depending on what is electron acceptor used in anaerobic process. In most cases, bacteria can only use one terminal electron acceptor. So if nitrate reducing condition, if nitrate is available, they use nitrate. When nitrate is completely gone, then there's going to be a transition of bacteria, then go for next electron acceptor condition available in the site, like sulfate or ion. So only one dominating electron accepting condition most of the time. That's what work in real world. Okay, and then we have facultative organisms that could use oxygen as well as other nitrogen, oxygen-containing salt, such as nitrate and sulfate. Here is bacterial metabolism aerobically and anaerobically. You can use oxygen electron acceptor, and you can use co-metabolic condition using additional carbon source. And anaerobically, various salt, you can use nitrate, manganese, iron, sulfate, and methanogenic condition, carbon dioxide could be used. So this is what pictorially represented in this slide. So here is your the L-NAPL from your underground storage gasoline tank. When it get into this groundwater, the first organism that use up is aerobic organism, and all the oxygen is gone. Then you can see the second group organism that take over is nitrate reducing condition. then iron reducing conditions, sulfur reducing condition, methanogenic condition. This is the natural order of redox potential. So this is how it works. So when oxygen run out, then you have the nitrate is used to electron acceptor, then iron, then sulfate, and carbon dioxide, which is the methanogenic condition. So that's the order of redox condition. So here is from the literature, you can look at electron acceptor conditions with different rank, 1 to 4. 1 is highly biodegradable, 4 is not biodegradable. So here's some of the compound solvents, BTACs, PCBs, and chlorinated compound like PCE and TCE. You can see that aerobic condition most of the time is highly biodegradable, but you can see under anaerobic conditions, so acetone can be degraded, and this TCE and PCE can be degraded using anaerobic conditions. And then BTACs also. Bioremediation practice, basically, you need to make sure the physical and chemical characteristic of the site, you need to understand what kind of condition it is. If it is, for example, clay soil, then you're going to have a serious problem because clay is going to absorb a lot of these contaminants. So it's not going to be available for bioremediation. You need to use some pre-treatment to make it available. It'll be more expensive. And you have to understand the complete pathway of what are the players, microbial players in the contaminated site, what kind of activity they do. And they, again, understand the environmental condition. And once you know that, then you promote these organisms, promote this condition, and then that's what this engineering aspect of cleaning up is, okay? So this is just basic fundamental stuff. Once you know each site what you need, then you go and provide that to the particular site. Again, oxygen is the primary source. Most of the contaminated sites are cleaned up by aerobic process because of the aerobic bacteria are faster than anaerobic bacteria. And here's a typical example how much oxygen needed for typical, benzene toluene compound, and what product you get. Everything is converted to carbon dioxide and mineralized. Okay, so in most of the cases, oxygen is all you need to supply, and everything else is in place. Okay. So how do you deliver oxygen? If you don't have it in the site, you can use hydrogen peroxide, pump the hydrogen peroxide below ground, and you can aerate your water and pump the aerated water instead of delivering oxygen in the gaseous form. And you can do passively using membrane, and then you can also aerate using pump to aerate. So those are commonly used method. Okay. So in a gaseous form, and usually when you use air, you use bioventing process. I mentioned in the last class. If your contaminant is above the water table, between the surface and the water table, it's in between there, above the water table. You use air sparging when the contaminant is below the water table, moved below the water table. So these are some of the common methods used for aerating the site. Okay. Then the dehalogenation reaction for chlorinated solvent, if a compound is highly chlorinated like TCE, you're going to use stripping the halogen and put hydrogen in its place. So here is a typical reaction of halogenated compound. The chlorine is removed reductively, and you put in chlorine place, you put a hydrogen. So this is called dehalorespiration and then anaerobic conditions. And you can also use co-metabolic conditions. So here what we are using is this TCE itself as electron acceptor. So you're just replacing chlorine with the electron. So dehalorespiration in chlorinated contaminated site is very commonly used. As most of the time, it ended up TCE converted into ethane, which is harmless product. And we can enhance this by co-metabolic process using EC carbon. You can have more bacteria involved in this process. High percentage of electron donor goes towards dechlorination. Okay. This Dehalorespiration depend on hydrogen-producing bacteria to produce hydrogen, because you need this electron, which is the preferred primary substrate. Okay. So this is a typical example. Here is your microorganism. Chlorinated solvent itself is the electron acceptor. You give electron donor, so it's going to consume, and then you replace this chlorine with hydrogen. So here's a PCE. You can see one chlorine at a time is replaced, and this is called reductive dechlorination process. Until you get to ethane, all the chlorines are replaced, and then chlorine becomes chloride, and then it precipitates out, it becomes harmless. Okay, so and then sometimes the reaction stops depending on the condition. For example, PCE can be converted into TCE, and the reaction stops. TCE is still very toxic, equally toxic as PCE. Okay. So that's what I just talked about. So if the reaction stops from PCE to TCE, you're still having toxic chemical in your contaminated site. So this is more hazardous than the parent compound. So when same thing with TCE to DCE, DCE accumulates. DCE can be 50 times more hazardous than TCE. And sometimes you get vinyl chloride from TCE metabolism, which is carcinogenic. So in anaerobic condition, this happens, then you need to promote aerobic bacteria to get rid of this vinyl chloride. So that's why you need to understand the whole process of what is going on in the site. So you cannot have just simply monitoring parent compound and say, yeah, I cleaned up site. That's not going to work. You need to make sure you show what happened to that compound. Do the whole mass transfer and complete mass balance, what happened. And then the co-metabolic process, it's a food-based transformation process because the organism that is using other carbon source, the same enzyme could also convert the contaminant. So, so this is commonly used. But the problem is when you have this additional carbon source, you're promoting a lot of the other organisms that are going to use this carbon source, but it's not going to do anything to your contaminant. So you have this problem of so promoting the right organisms, but not promoting all the organisms. We call that weed bacteria, like a weed. So you wanted to make sure that you promote the organisms that you want, that carry out your particular dechlorination, dehalogenation reaction. So here is again the example of selective enhancement of reductive dechlorination. The competition for hydrogen is in subsurface is big because most of the time the hydrogen can be used by other organisms like methanogenic bacteria can use the hydrogen to convert them to methane gas, sulfur-reducing bacteria. So you need to have this hydrogen partial pressure and hydrogen balance, and you want to make sure this competition is not going to inhibit your dechlorination process. So that's another tightrope you need to walk when you're doing this kind of cleanup. So what kind of electron donors you could use? If you're using co-metabolic conditions, you can use a lot of alcohol and acids, fatty acids you can use. A lot of fermentable compounds you can use, like molasses and corn syrup, and anything that is cheaper you can use. Hydrogen is apparently your electron donor, so hydrogen can come from a variety of organic substrates. So that's why you need to do a small-scale field study, small-scale studies before you actually do which donor is going to work for you in the field, electron donor. So here is a variety of donors commonly used, anywhere from acetate to molasses to a lot of different compounds like cheese, some of the base from chicken manure, cheese whey, corn stick, liquor could be used. And then you can use some fine chemicals you can use depending on how expensive or how cheap you want to do your cleanup process. So there's a lot of electron donors commonly used to do the cleanup. Just to go through the enhanced bioattenuation process, You can do the petroleum hydrocarbon chlorinated solvents, the electron acceptor, electron donor category. So you can use aerobically or anaerobically, depending on your site. And how you do this, biospa using oxygen, slow release oxygen you can use aerobically. And anaerobically you can do that also. Then chlorinated solvent cleanup, most of the time the electron donors are, as I said, benzoate, lactate, and molasses. Molasses is very commonly used because it's cheap compared to other carbon sources. Okay. So those are, I give you a general case studies for heavy metal for phytoremediation, using phytoremediation, and then TCE and oil spill cleanup below surface. So this is, a lot of people have done this, a lot of success stories that I'm just give you a general overview, okay? And some limitation in the process of like competition for electron acceptor process. So now I'm going to talk about some specific cleanup that I was involved with. I had some research project I conducted in cleaning up a particular explosive contaminated site in Louisiana. I'm going to share some of those results with you. And then I'm going to talk about the coastal oil spill that BP oil cleanup in 2010 in Louisiana, which I was involved also. So now I'm going to talk about this specific cleanup case studies, okay? So this particular one is talking about cleaning up of explosive contaminated soil in this particular ammunition site in the town in Minden, Louisiana. So this TNT was considered as recalcitrant because this compound has been sitting there since Second World War, from 1930s. During the production of Second World War, this compound has started produced in that site. But the compound is still there at a high concentration. Nothing happened to them. So they classified as recalcitrant. And they are recalcitrant because of the unusual substitution. If you look at TNT, it has 3 nitro groups in its structure of toluene. So toluene could be easily degraded, but when you added the nitro substituent, the bacteria have a hard time, degrading that compound. So when you have this unusual substitution like chlorine and other halogen or nitro groups, and then it becomes recalcitrant. And then other reasons also when you have high molecular size like plastics. Again, why the bacteria didn't do the cleanup? Because the enzyme was not synthesized. The compound itself either failed to enter the transport process. There's not suitable permease to transport them into the cell. The compound is unavailable due to the limitation of solubility. TNT maximum solubility is only 100 ppm in water. And then when you have high concentration of the compound, it's going to be toxic to the bacteria. So these are some of the reasons why this compound persisted for a long time. So if you look at bacterial metabolism, just Biochem 101, you have this metabolic pathway in bacteria. Normally they use glucose, and so when they do that, they do this glycolysis, and then they convert them to acetyl-CoA, and then the acetyl-CoA go to Krebs cycle, and then the electron transport system, where when the electrons are transferred from one electron carrier to another electron carrier, bacteria gets this ATP. So from one glucose, you can get 38 ATP molecules. And when you have long-chain fatty acid, what they do beta oxidation pathway, which is chopping 2 carbon at a time, converting them to acetyl-CoA and take the acetyl-CoA to Krebs cycle. So bacteria is not inventing any new pathways. These are the pathways through the long evolutionary process the organisms have evolved into. So when they see this new chemical that we throw at them, what they do, they modify the structure. So they make some enzyme like dioxygenase, monooxygenase to break the ring. And then once they convert them to either an acid or alcohol, then they shunt those metabolites into these existing pathways. So this is the simple, basic mechanism of microbial physiology. So they always use the existing pathways, so they never create any new pathway for the new chemical we're throwing at them. All bacteria does is modify the compound and then try to fit them in any one of these pathways, okay? In most of the time, the cleanup in real world is not pure culture, it is synergistic degradation. So there are 2 or 3 groups of organisms. They do, one group of organisms do biotransformation and then convert the compound to some metabolite. Another group take them to next step until it's completely mineralized. So you need to have more than one group of organisms doing this reaction until you eliminate it. So very rarely you see pure culture in cleanup process. Most of the time it is synergistic. You need to have mixed culture of bacteria in cleanup site. So this particular site I'm talking about is contaminated with this 3 explosive compound, TNT, RDX, and HMX. So if you look at it, these are all have this nitro group, make it very unusual substituent to this molecule. That makes it hard and that makes it persistent in the environment for a long time, okay? So this is a trinitrotoluene, this is hexahydro 135, trinitro 135 triazine, commonly called Royal Demolition Explosive or Rapid Detention Explosive, the 2 names, that's why it's called RDX. It's commonly known as plastic explosive. Then we have HMX, which is high melting explosive. It's the octohydro 1357 tetranitro 1357 tetroazosine. So these 3 chemicals persisted in this soil for more than 75 years, never went away. And this is this particular site. So I live down here. This particular site is up in north part of the state, and the town is Minden. This particular site is more than, close to 16,000 acres. It's been contaminated since 1941. This is a big area, and there are a lot of production sites. When it was highly active, there are 8 factories right there in the site that produce this various bombs, chemical. And then they stopped production in 1994. And now currently it's because of this contamination, they fenced off this area and in a caretaker status. So the Army by law has to clean up the site before they use this site for any other purpose. They can hand over to local government, but they cannot hand over until they clean it up completely to the regulatory standard. So that's where this site is right now. So we got involved in doing some research in this particular site to look for how to clean up this site. So what we did review the existing data. We did a very extensive physical chemical analysis of the site, collected sample, brought to the lab, we characterized this sample, and we did small biotreatability study, and then we assessed, and then we did a bioremediation study, and then we recommended one method. So this is what any bioremediation company, when they do the cleanup, this is what they do. They do look at what kind of data is already there in the site, and then you collect sample and go through this process and then finally execute the cleanup. So our objective is to look for first, why these chemicals are there for such a long time. Are there microbes inside that can clean up? Are the microbes are not there? Okay, that was a very basic question. So if the microbes cannot use this compound, can we use this electron donor I just talked about, provide some molasses to produce more bacteria? Maybe they will pick up these compounds. So those are some basic, simple questions we asked. So we went to the site, collected, we characterized, we collected extensive soil sample, and they did the analysis just to show HMX in HPLC, come at 3.3 minute retention time, RDX come at 4.8, and TNT comes at 10.69 minutes. And then when you extract the soil, and you can see all 3 of them, So the HMX, RDX, and TNT. It also has other residues. Some of them are bomb chemicals, some of them are metabolite. But we are concerned about these three chemicals and how to clean up these three chemicals in this particular site. So this is a almost 16,000 acres. So if you look at the TNT contamination, it's 4,000 to 10,000 milligram per kilogram of soil. So the lowest range was 4,000, the highest was 10,000. And the RDX is 800 to 1,900 milligram per kilogram. HMX is 600 to 900 milligram per kilogram. This is the actual concentration of these explosives in this soil, right? The Army wants to bring this to below 200 and the RDX to below 50, HMX to below 20. So that's their goal before they can handle. So first thing, you ask a simple microbiology question. We put the soil and enrich the soil, transfer it, isolate it, pure colonies, basic microbiology. And then we use the different conditions. These are some of the media. We use basic mineral salt media. And then we have another condition, co-metabolic condition with glucose in the lab. And then once you isolated, we stored the bacteria and then did one by one experiment. Okay, we isolated 22 pure cultures. We screened them all, each one of them. It's a very tedious, time-consuming process. And I'm going to just share results with maybe 2 bacteria. It's no time to share all of them. So Staphylococcus is one of them, and Bacillus levolacticus is another one. We grew this pure culture in different conditions. So we had obviously control, no bacteria, and then we had used the TNT as sole carbon source, no other carbon in the flask. And if you look at TNT has nitro groups, so you could use TNT as a nitrogen source for bacteria. So we eliminated the ammonium salt from the media. The only source of nitrogen is the TNT. And then we use co-metabolic condition where we had glucose and the explosive together. So these are the various conditions we use. Then we monitored the growth and we analyzed the compound in these cultures. So here's quickly to go through. So if you look at staph, various growth conditions, control, carbon source, nitrogen source, co-metabolic conditions. You can see the growth was better when you have additional carbon source with glucose and explosives. And then when you use them as nitrogen source, the bacteria use that TNT as nitrogen source. Somehow they kind of remove the nitro group and use the TNT as nitrogen source. And then the carbon source also bacteria grow on it. Okay. The best growth obviously was co-metabolic conditions. Okay. And then the degradation of the TNT, you can see the same thing. The co-metabolic condition was faster than carbon source conditions. Okay. Same, almost same. The trend for Bacillus, various conditions, the co-metabolic worked the best. Co-metabolic removed the TNT the best. Okay. But when you use the other explosive, the story is different. RDX cannot be used as sole carbon source. When you use RDX as sole carbon source, nothing happened to the culture. Only co-metabolic conditions showed growth, and co-metabolic conditions removed the RDX. You couldn't use a nitrogen source, carbon source. Both organisms did the same, staph and Bacillus. So the take home is TNT, if you provide the right condition, bacteria could use them either as carbon source or as nitrogen source. But of course, if we had an additional carbon source as co-metabolic, it was faster. But the other 2 explosives, the RDX and then the HMX did the same thing. HMX, only co-metabolic condition worked in both cases. It is the same story for 10 of the isolates. Okay, other 10 isolate worked a little bit with HMX and RDX. Okay, so this most simple enrichment study and isolated pure culture with each one of them showed a general trend. TNT, if you put the right condition, can be degraded without any trouble. For the RDX and HMX, though, you need to provide additional carbon source. Otherwise, it's going to persist for a long time, which is what's happening at the site. Okay. So this is a simple degradation study. Then we looked at what kind of method will work, whether you need to use land farming, soil slurry reactor, composting, phytoremediation, which method to recommend. So we looked at the literature, all the different methods I just talked about. Again, to revisit bioremediation definition, so we want to intentionally use the degradation process, so we enhance the microbial activity. We, again, same protocol for bioremediation, go through different existing methods, and then which method to use for treatability study. So we chose these two methods, the soil slurry reactor. The reason for that is the method is fast. You can remove the contaminant really, really fast, but the disadvantage is capital intensive. It's going to cost you more money. Okay. The land farming has a potential for significant savings. The operating cost is less, but the reaction time is slow compared to. So we want to compare these two and give the Army an option, which one to use. Okay. So we set up a small soil slurry reactor. We had a control. There was no molasses added. And then we had treatment. We had molasses added. We had a slow mixing. And this was with 20% slurry. You put soil and make it slurry with water. All you add is just the molasses to the treatment and no molasses. That's all. We didn't do anything else. Then we just periodically took sample and analyzed what's in the soil. So here's the average of those two, treatment and control. So when you had molasses added as co-metabolic conditions, you can see that the TNT concentration going down really fast in the red line. And when there was no molasses, it's going down, but it's very, very slow, right? So adding molasses, 0.3% by volume, really helped. The TNT went down to the recommended level within 6 months of the reactor operation. So the Army wants to bring it to below 200. We did bring it to below 200 in 6 months. Surprisingly, the RDX also went down because it's again, it's because we're enhancing not pure culture. In the pure culture, it didn't work, but in the soil, adding molasses helped to bring in a lot of microbial activity. So it took a long lag period. Nothing happened to the RDX for 2 months. After 2 months, RDX in the soil went down in the reactor with molasses. In the reactor without molasses, just hang around, and nothing happened, right? So co-metabolic condition, the RDX went down after 2 months of reactor. We were kind of, first 2 months, we kind of, whether to stop the experiment or keep going. But we said, let's go for 6 months like the TNT, and eventually it went down. Okay. And HMX is also took a very long time before you can see any decrease in concentration. So without molasses, it just persisted. And with molasses, it took almost 3 months before you can see big drop in HMX. So this small, simple study tell us that by putting molasses and mixing, you can remove these 3 chemicals in a slurry reactor within 6 months. Okay, that's the result from this study. And then we want to do the land farming. We started out in the small scale. So just to simulate the land farming process, in this pan, we put the contaminated soil. And in this pan, we had molasses. We just flood with molasses, and then we drain the molasses. And then we till the soil by aerating the soil. In the control, no molasses, just the water. right? 0.3% molasses in these two pans. No molasses, just the water here. Okay. Again, the same kind of result. When you had molasses treated land farming pans, you can see this TNT go down, but it was not fast. It was not as low as soil slurry reactor. Soil slurry reactor went down below 200, but it did go down in the land farming, right? Because you can tell the land farming is just a solid phase system. Whereas in slurry reactor, you make it a slurry mixing, so you have better mass transfer. So the reaction was faster there. Here, reaction was slow. So, but you can see this eventually went down significantly. Same story with RDX. It took a long time in the land farming pans. It started to go down. Okay, after 4 months in land farm. And HMX also. It took a very long time for HMX to go down, but HMX eventually also started to go down. So what this tells us, if you have a lot of time, you don't want to have somebody holding gun on you to clean this site right away, you can use land farming. This will take more, maybe 2-3 years to clean it up, but it eventually will clean up these chemicals. So that's basically what, but you need to apply this molasses every month, once a month in the field. So the summary of these two bioremediation, soil slurry land farming, you can see this removal of this chemical, 99% in TNT in slurry versus 82%, RDX 97 versus 90, HMS 87 or 71. So both of them removed, slurry reactor removed better and faster than land farming. Okay. So now all I showed was just the removal of the compound. Now I had to do metabolic pathway and make sure there is no activation going on, make sure all these compounds are converted to harmless product, right? So we did the mass balance study by spiking radio-labeled TNT and then monitor the radioactivity of C14 TNT. And you can see in the control, most of the TNT is still there. And you see some metabolite, these are the biotransformation product of 4-amino dinitrotoluene, 2-amino dinitrotoluene. They are produced in the control. But when you have molasses, you can see this. 22% of the TNT is mineralized to carbon dioxide, which is spectacular, that 22% mineralization. And 23% of the radio-labeled TNT ended up in making more biomass. Bacteria use them to make more bacteria in the cell. And then we have a lot of other metabolite accumulating, and but these two account for almost 50% of CO2 as well as accumulating in biomass. Almost the same story for land farming. Most of them without molasses remained in the soil. Nothing happened to the compound. But when you add molasses, you produce again CO2. You produce the TCA, which is the biomass precipitable material. Okay. So this tells us that TNT is mineralized, and this is a consortium of bacteria, not pure culture. The pure culture only, I showed you a few pure culture, but this one has a lot of bacteria in the soil. All the organisms are enriched, and they are doing a fantastic job of mineralizing this explosive. So this is the kind of aerobic process because you're mixing, your oxygen going in there, and then you're also land farming, you turn the soil over. So you're actually increasing the aerobic bacteria. We want to know what is happening in anaerobic condition, because if we take one single soil particles, you can find oxygen gradation. The surface of the particle is aerobic. As you go into the middle part, it is anaerobic. So if you can promote anaerobic activity, we want to know what kind of TNT degradation happens in the soil. So we promoted sulfur-reducing bacteria by providing 20 millimoles sulfate And then we developed a consortium of desulfur Vibrio species. There are 4 different bacteria, and the consortium used TNT as sole carbon source. So there are anaerobic bacteria in the soil. So, so if you provide some sulfate, and the bacteria will pick up this TNT as sole carbon source. So here is those consortium we use in the lab. This one is with the co-metabolic condition. We use pyruvate as substrate, co-substrate. And then this one is without pyruvate, just the TNT. And this is just a control without bacteria. So you can see the growth, and you can see the TNT go down. So co-metabolic, of course, went fast. And sole carbon source, like 4 weeks almost before it went to 0 in the culture. So this tells us that if you, as I said at the beginning, you can provide some electron acceptor, so like nitrate, sulfate. So oxygen, of course, is good, but if there are some anaerobic pockets. So if you want to encourage that bacteria, if there is some sulfate already there, why not you enhance the bacteria? So that's what we did here. And then we created a slurry reactor, anaerobic slurry reactor, and enhanced the activity and monitor TNT. And you can see in the slurry reactor, the 6,000 milligram per kilogram of TNT went down within 100 days in the anaerobic conditions. And the control when there is no bacteria and nothing happened to this TNT. So there's no abiotic activity, mostly biotic activity that's taking over anaerobic process. Again, we did the radio level study. We spiked some radio level TNT and accounted for complete mass balance. This particular anaerobic bacteria is what we call is incomplete oxidizer. It doesn't take the organic compound all the way to CO2. So what it did was if you look at this column, the radio label TNT 27% of it converted to the cell biomass. Most of them are converted to acetic acid. So the acetic acid accumulated in the system. 47.5% of TNT is converted to acetic acid. This particular consortium doesn't have the final step to oxidize acetic acid to carbon dioxide. So that's our end product from TNT to acetic acid. Then we produce these are some other metabolites, nitrobenzoic acid, butyric acid, pentanoic acid, and these are biotransformation products. So based on this, we constructed the metabolic pathway. So this is what's going on anaerobically. So here is your TNT. The thing happening here is this transformation, reduction of the nitro group to amino group. So this becomes 4-amino 2,6-dinitro. And then in the second position, nitro group is reduced. This has become 2-amino. And then 2 and 4-amino reduced become 2,4-diamino. And then we couldn't catch some other metabolite. The next metabolite we caught was this nitrobenzoic acid. So we are kind of suspecting this amino group is cleaved. There's a deamination reaction. This amino could be used as a nitrogen source for bacteria. And then the methyl group is converted to carboxylic acid, and we found nitrobenzoic acid. And then after that, the ring cleavage. And once the ring is cleaved, then you have cyclohexanone and then fatty acids. Once you have fatty acid, then bad oxidation pathway, 2 carbon at a time chopped and converted to acetyl-CoA that goes to Krebs cycle. And then all the way, everything is converted to acetic acid. So you have pentanoic to butyric, propionic, and acetic acid. So this is the end product from harmful TNT to harmless acetic acid. Acetic acid could be used by aerobic bacteria as a carbon source, right? So this is what's happening anaerobically in that soil. So that's particular site, we recommended 2 choices we gave the Army. You could use a soil slurry reactor if you want to do a fast cleanup. If you have a lot of time to clean up, you use landform method. There's a trade-off between the time versus the cost of cleanup. And what they did was they divided the site into several areas. the immediate area that they want to hand over to the local government, they did the slurry reactor. And then some area that protected for nature conservancy project, they went with the land farming method. So that's what that site has been cleaned up. Okay. I'm now. Can I continue with one more case study, Tjandra? You have
Please.
time?
Yes,
okay.
Still time. Yes.
We have still time. Okay.
Yes.
So this is, I was involved for real cleanup, and we did the lab study, we did the treatability study, and we recommended the method. The method has been executed, and out of the 15,000 acres, I was last told half of it cleaned up, 7,000 acres being cleaned up and handed over to the local government. Okay. So now let's talk about the oil spill. Right. This, some of you heard about this 10 years ago, a little over 10 years ago, on April 10th, 2010, we had a big oil spill right in our backyard. I live 75 miles from this actual site where this BP oil blew up. So this was in the Gulf of Mexico, and some of the history behind what happened is this is what happened. The explosion happened in Deepwater Horizon drilling platform. 11 people died. And it took until it happened in April until September 17th to stop the oil, to cap the oil. And it took a long time to contain. And during that time, we had 50,000 to 60,000 barrels of crude oil flow happen every day, per day. Okay. It covered a wide area, 2,500 square miles. And by the time they capped the well, there were 170 million gallons of oil spilled in the Gulf of Mexico. This is the largest marine spill in US history, okay, 10 years ago. More than 50% of them were dispersed at the depth, like 10,000 feet below, 5,000 feet to the surface and then below, okay? So during the time, they used 1.8 million gallons of dispersant, which is commercial one called Corexit 6500. That was the dispersant they used from Nalco Chemicals. They used the dispersant at the site where the oil is coming below the seabed, under the seabed, and also used aerially. They applied an open ocean. So as a result, a lot of things happen. People are affected economically because all the fisheries shut down. A lot of people lost their livelihood. Environment was severely damaged. The whole state suffered economically. And then we had geological problems in the Gulf of Mexico because of this oil wells problem. Some of them are chronic problems, some of them are acute. So most of the acute problem is gone, but this chronic problem still persists. People still have this effect in here. Okay. I just want to make a point that in Gulf of Mexico, we have a natural seepage of oil. In the seabed, every year, we have 50 million gallons of oil naturally comes out. Okay. That's part of the natural process. Okay, but all of this oil came all at a sudden time, all at once. That was the problem. So when it's slowly leaking, microorganisms take care of that. Okay. So first thing what they did was they want to kind of on-site, in situ burning of oil. So whatever oil that accumulated on the ocean surface, they burn it up. So they light up and they use some accelerant to burn the oil. So you can see this big plume of burning of this crude oil. And then they contain it. They want to contain it. They use these big skimmers and containers to contain this oil getting into the shore. Okay, they use mass evaporation. BP spend billions and billions of dollars. So this is how the site looked like pictorially, schematically. So this is where they drill. This is the sea surface, 5,000 feet to the sea floor. And then they went 13,000 feet drill. to reach this oil. All right, so that's where this oil, Makanda seawater. So in the Makanda reservoir, the problem was it has high methane content. And there was some problem with this automatic shutting valve that they were not properly taken care of. So this methane built up and the methane blew up. And as a result, we had a big explosion. And then the whole thing, there's no containment. So that's what happened. Okay, so a lot of methane in this oil, natural gas. So here's some picture to show you the damage. You can see oil on the surface, on the shoreline. Most of the shore are wetland, salt marshes. Salt marshes are covered. And this is the Gulf of Mexico. Here's the Florida. We have Louisiana right here. And this happened here. So the current took the oil everywhere. It went all the way to UK, Atlantic. The loop current took them all the way to UK. See the damage, a lot of wildlife were killed and a lot of oil on the coastal marsh area. And then we can see the activity on the surface of the ocean, okay? So 7,721 miles of coastline of Louisiana was affected. A lot of them are this marsh wetland. We have a lot of wetlands, highly productive wetland, very good fishery spots. So all the fisheries shut down. They cannot fish this area. And just to show you more pictures, what they do, they constructed some sand berms to catch the oil so it won't reach these fishery spots. And they put this also the booms to contain oil. And this is what's happening. And you can see the damage when they do this kind of activity to contain oil. Okay. So it's more pictures to show you. They have small barrier islands. They cover the whole barrier islands with this boom to make sure it won't reach, because these are a bird sanctuary, migratory bird land here. They want to protect this, and they applied this dispersing agent, right? Open ocean they applied, okay. So just to, if you look at the date, there are different days. There's a June picture and it's a July picture. It looks like no oil, right? But when you go close and take sample, the oil is still at the bottom. You can see it. Within a month, most of the volatiles evaporated, and some of the wave action took the oil back into the ocean. But there is, I'm going to show some close picture, but still some oil after the July picture. So you can see a lot of those local ecosystem. This is oyster. This is where they harvest oysters, and they're all completely covered with oil. Actually, they can't use them at all. For a long time, we couldn't harvest oysters. So you can see this, the area that I showed you from long shot. You don't see it, but when you actually go close, you can see the oil still there. Right? In September, it was still there. You can see the initial oil, and then gradually you can see the mark everywhere. Okay. More pictures just to show you the damage to the ecosystem. A lot of oil in the shore, and that's your wet marsh over there. This is how it looks like in close. And this is the dispersant. Without dispersant, when you don't have dispersant, after you apply dispersant, the oil is dispersed. Okay. Of course, dispersant has its own toxic effect on the ecosystem, but at least it broke the oil into smaller particles and dispersed. Okay, just to show you what happened in a picture. So when you have oil like this, when you add dispersant, it actually dispersed out. It'll be clear within a few hours, few minutes. The dispersion has a really very good effect on oil. So what happened? The mass balance of oil, according to NOAA, all the 170 million gallon of oil that spilled, 17% DP recovered right at the wellhead. They sent some big pipeline to catch the oil. And they 5% they did the open air burning. And then a lot of small boats went and skimmed the oil using some pump. So seawater and oil mixed together, but then later they separated it. Then chemical dispersion using this Corexit, it dispersed into open ocean and went into this loop current into Atlantic Ocean. 16% is naturally dispersed because of wave action. 85% is either evaporated or dissolved in the ocean. But we still have this 25% oil in the coastal bottom. So every time there's a big hurricane or big wave action, this oil can, come back. So this is what they're worried about. This 25% is going to have this, what do you call that, chronic effect on the ecosystem. So this is where we come in. So how are you going to get rid of this 25% that is buried in this coastal ecosystem? Okay. So our research project was to help the oyster farmers, because the oyster industry in the Southeast Louisiana is big. And it's a small farm, not a big operation. These are all small individual oyster farmers. So it's a good industry. In New Orleans is famous for oyster seafood. And so we went into these two sites where the oyster is. We collected the sample and brought to the lab and did our basic studies. Okay. So this is how it looks like when we went to take sample. It looks pristine. It looks clean from a distance. Okay, when you go and collect sample at the bottom, because it's all at the bottom of the coastal area, you can find oil. And this is oyster sample, and then we collect the sediment, oyster, and water. And we did the analysis of oil, and then we did the enrichment study for bacterial activity and how the oil can be cleaned up. So what we did was we did the redox condition of the sediment. The sediment is not aerobic, but sediment has a lot of nitrate and sulfur reducing bacteria. If you just take 1 inch of the sediment, you can smell the hydrogen sulfide. So there's a lot of sulfur reducing bacteria there. So if you put enough sulfate as an electron acceptor, maybe this bacteria will take care of the oil, right? So we set up various conditions: nitrate reducing, sulfate reducing, ion reducing, methanogenic, fermenting, and then we put all of them together. We call it mixed electron acceptor. We want to promote all the bacterial activity in one condition and see whether that will do anything good, okay? We set up a soil slurry reactor, and then we did a soil column study. We took a core sample, and then we mimicked each of these conditions in core sample. So these are the slurry reactor conditions, and we had a duplicate culture bottles. Because of 2 sites, there's so many bottles, so we couldn't do triplicate. And then every sampling event, we sacrificed each duplicate bottle, and we did the total petrol hydrocarbon. So we set up the condition, and then we took the whole bottle for every week, and then we did the analysis. We followed the EPA protocol, the experimental setup showing different culture conditions in a big shaker. Quickly, the result, we monitored the total petroleum hydrocarbon. We didn't care about analyzing how much is aromatic, how much is aliphatic and polyaromatic. We just did the one total petroleum hydrocarbon. All the total area in a GC was taken. And you can see when you have the mixed condition, when you have all the electron acceptor, the degradation was better. It was surprising. I thought at the beginning of the lecture, I told you, right? Generally, microorganisms go 1 electron acceptor at a time. This is totally opposite. I said oxygen first, nitrogen reducers come, then ion reducing bacteria. But in this case, when we put all of them electron acceptor, that condition removed oil better. And then you can see this blue line. The blue line is the fermenting condition. That means no electron acceptor. It's just anaerobic, we want to make sure the oil itself could be used as electron acceptor, that so it can degrade and put the electron in the oil metabolites. Okay. And this, of course, control no electron acceptor at all. And then when you put individual electron acceptor, the sulfate reducing condition, so out of the mixed electron acceptor condition, it looks like the sulfate reducing bacteria is doing the bulk of the job. So when you have only the sulfate, you can see 55% oil removal. And when you have nitrate, you have 30% oil removal. So if we combine these two, it accounts for the mixed electron acceptor condition at 80% oil removal. So this looks like there is some kind of synergistic activity between these two groups of bacteria in this sediment. And then based on this quick study of 4 months, then we set up this core soil column experiment where we put several soil columns and mimic the same conditions. This soil comes from the site I showed you. We dig up the core sample and put it in there. All we did was we pumped sulfate, 20 millimole in sulfur reducing condition, nitrate reducing condition. We put nitrate and mixed condition, put them all together. So quick result, we can see the CO2 production in the, I'm only showing the mixed condition. You can see the CO2 production going up really well. We dose it and based on the wave action. So when there is the high tide, low tide, and wave action, we drain the column. So you can see no CO2 when we drain it, and then when we flood it, the CO2 comes out. So microorganisms are working on this oil and converting them to carbon dioxide. And then we also monitored methane. So you can see the methane compared to control and mixed electron acceptor condition produce more methane, okay, and different periods, right? And then we did the count, how many bacteria are there using this, some kind of gross analysis of making the media to promote only this kind of group of bacteria. So it's not very accurate, but it just gives a ballpark figure of what is going on in this condition. So when you have all the electron acceptor, that's where you have a lot of bacteria growing in the soil column. When you have this, the blue line, which is the sulfur reducing, is the second best. And then you have the green line, the methanogenic condition, and then you have the nitrate reducing condition, okay? So this is the order. So when you have all the electron acceptor together, you have better bacterial growth in the soil column, and then you have sulfate, methanogenic, and nitrate reducing condition. And just to show you what happened in the soil column, at the beginning, you can see a lot of this crude oil fraction in the column. And then after 160 days, you can see the same soil column, mixed electron acceptor condition. You can see the signature peaks coming down. After 290 days, you can see almost minuscule amount of carbon left there. And this is the removal activity in the soil column. And you can see mixed electron acceptor worked better than the slurry reactor in the soil column, worked better than slurry, but the operation was almost, 550 days compared to in the slurry reactor result was 120 days. So the carbon removal was really good in mixed electron acceptor condition. Then he went down sulfate and nitrate reducing conditions. So, and just took the best condition, we put this bacterial growth, we put the percent removal, the mixed electron acceptor condition. You see the mimicking bacterial growth with the highest removal activities. When they're actively growing, when they're dividing cells in the soil column, you see the best removal activity. So, and then it reached the point where most of the carbon is gone, and you can see the bacteria started to go down. The number of bacteria going to go down, like 2 orders of magnitude down, going down. So this just tells us that if you provide, you don't have to provide all electron acceptor. If you provide at least sulfate and nitrate, there are already there, some of it, but according to our study, you need to have 20 millimoles. So we need to kind of formulate this to add this to this site. Maybe this will speed up the actual cleanup of those 25% oil in the coastal sediments. Okay. So I just took some electron microscope. You see a lot of bacterial activity in the soil column, all kinds of different heterogeneous populations. So this study basically showed us that, again, at the beginning, I said the electron donor, electron acceptor, you need to know which one to add. And if you find the right mix, the bacteria will do the job. That's what we did here. So the nitrate and sulfate reducing condition worked better for us. When you add both of them, mixed condition worked really, really better. So there was a little bit of surprise to us. I thought one condition worked better than other, but mixed condition worked really well. So, so the funding for this work, both of the study come from Department of Defense and Army Corps of Engineers and National Science Foundation and Louisiana Board of Regents. So that's two case studies I was personally involved. I just sharing the data with you. So I will stop here, and I'm glad to take any questions. Yeah.
Okay, thank you, Raj.
Right.
Probably there are some question, right? Probably.
Okay.
You stop.
You want me to stop sharing this one?
Yes.
Okay.
Yes.
Okay.
Okay, Guntur.
Yes, Pak. Wait a minute. Just a minute.
Okay.
All right. So you want to go through the first question?
Okay. So
yeah, it's talking about how to supply oxygen for aerobic in situ bioremediation. I was briefly talking about both active and passive. You can actively pump hydrogen peroxide. You can actively pump aerated water into the system. Or passively, you can do, pump air. But as I said, it depends on where the contaminant is. You can do between bioventing and air sparging. Bioventing is above the water system, and air sparging is below if the oil is below. So depends on the site. Yeah.
Okay. And it's the second one.
All right. What is the best method to introduce bacterial consortium to degrade biomaterial waste? If they need different conditions, should we manually add them one by one, or we add them together in the beginning? Well, as I said, you need to do a little bit of lab study first. Sometimes the nature throws some surprises, like I showed you the last experiment we did. The mixed condition worked. We put them all together. That worked better. We didn't expect that at all. So we thought individual electron acceptor worked better. So always it's good to do a little bit of bench scale study before you can apply this. So there is no one size that will fit for all the scenarios. So it depends on the site. It depends on what biomaterial you're trying to clean up. So.
Okay. So the third one.
The effect of dissolved oxygen and ammonia levels on the growth of indigenous bacteria and bioremediation are condemned by crude oil. The effect of dissolved oxygen and ammonia level. I didn't show you the lab study, the media and all that. So when we did this oil spill study, the aerobic conditions, Yes, oxygen really worked well. For our condition, you need to have anywhere from 2 milligrams per liter, from 0.2 to 2 milligrams. So we have like microaerophilic to aerobic conditions. So 2 milligram or less is okay, oxygen in the water. And then ammonia, you need to have, in our system, 30:1 ratio, carbon-nitrogen ratio. For every 30 parts of carbon, you have 1 part of nitrogen. So 30:1 ratio is what we used in our study. So, so you got to do first how much nitrogen is already there in the site, and then you work with that carbon-nitrogen ratio. Yeah. Any other questions?
I'm still mute. So
Okay.
is there any other question? Or probably Bu Reni, if you have any, please, Bu Reni.
Yes, thank you.
Thanks.
Yeah.
Reni?
Yeah, okay. Raj,
Yeah.
kind of blowout in Indonesia, not too big, somewhere near Karawang. Yeah, and they managed to get the spill stopped, but we do have a problem with mangrove.
Uh-huh.
Mangrove. Many of the plants were still contain some sludge, the oil sludge.
Okay.
And, what's the best way to cure the plant? I mean, it's not possible to remove because mangrove is very long to grow, and it's kind of a pity that they contaminated with the oil sludge. Yeah. Any idea how to treat plants that?
So is the mangrove plant still alive? They're not dead by this oil sludge?
Some of them they still alive. Yeah.
Okay.
One that's badly, yeah, we have to get rid of them, but we still have some that possibly they will survive. That I think we have to do.
So in our experience, do you have some salt marsh also in the mangrove? You don't have any salt grass in that area?
Oh, no. Yeah.
Oh, because the salt marshes really take up this oil very well. So, and if you have salt marshes, or I don't know whether you can plant, that will enhance your oil degradation process. And also it will promote the rhizospheric bacteria. So you have that, the site I showed you has some salt marsh. And the sample that we collected, the root of those salt marshes is where the bacterial activity are. And those are the one that was actually doing the oil cleanup. So you got to have some, help from the nature. Otherwise, you got to add them. Maybe promote some particular anaerobic bacteria. What is the redox in the sediments?
Mm.
What kind of redox conditions?
Yeah.
It's a wave action. You have to take into account the wave action. Every time wave comes in, the tidal action, it will oxygenate your sediment. So
yeah.
If it is kind of the zone between aerobic and nitro reducing, then maybe you got to promote that activities there. Yeah.
Okay.
Yeah. We don't know yet.
Yeah. There's a lot of those things you need to look into it. But you cannot just show, this bacteria from the literature seem to remove, maybe we have to do that. No, you have to look in your site. Okay, what kind of plants are there? What kind of redox condition is there? How much wave action in your particular site? Is it a shallow coastal area?
Yeah, it's yellow.
Yeah, okay. Yeah.
Yeah. Okay.
Yeah, I mean, it's then when we start discussing about it, then we have a pandemic, so we haven't done anything since.
Okay. So you are involved in that project. You got to collect some treatability study, collect some sample, look in what kind of activity you have in the sediments.
Mangrove samples just to find out to what extent the disturbance to the plants.
Yeah, if the sludge is in the sediment of the mangroves, you can look at the bacterial activity in that sediment, whether they have maybe sulfate reducers are there. If it smells hydrogen sulfide, definitely there is sulfate reducing activity going on.
So far, we focus on the mangrove itself. So
mangrove itself, yeah. But he said the mangroves are completely covered, and yeah. And you need to have other plant to help the mangrove. So mangrove are very, very susceptible to oil. Yeah. Very sensitive so for oil. Yeah.
Thank you, Raj.
Thank you. So I think this one question from me. I think the last one it might be. So Raj, you mentioned about the gas station. So I think the leaking from the gas station.
Yeah, underground. Yeah.
So that one definitely the gas station or the owner of the gas station cannot afford to pay for the cleanup, right?
If the owner is still in business, they have to do the cleanup.
Yeah, but
if the owner is gone, then owner is, you cannot find the owner, then the government will do the cleanup.
Oh, so the government will clean up, and then they have a, what you call it? They have a super fund.
Super fund, yeah. In EPA, every budget, every year, the government put the money in the budget called super fund, and the money is used for cleaning up the priority site. There are every year 1,300 sites are considered priority site. So they update every budget year what are the sites need to be cleaned up first. So they use that super fund to clean up those sites, and then they try to recover the money later. But if the gas station guys are still in operation, then the government will come and ask them, to clean up. But if they're not there, they're gone, then nobody knows where they are, then the government use that money. Yeah.
Okay, then I think so. No other question, Guntur. Is already.
Yes, there are no other questions.
Okay.
I think we may close our class, our session today, and then I will for the next weeks. So because you mentioned it might be the topic on the microbial lipid on bioenergy.
Next week is biological treatment of wastewater.
Okay, so your biological treatment.
The week after is microbial lipids and lignocellulosic acid.
Okay. Okay. Next week is biological treatment. Okay.
Biological treatment of wastewater. I'll talk about some basics and then I'll do some case studies.
Okay. I think we will close our. Arik, probably you can stop the in the YouTube. I would like to thank, and then next week we will have a lecture from Prof. Raj on the biological treatment of wastewater.
Wastewater, yeah.
Thank you, Raj. I think
all right.
So
thank you.
Say hello to Rose.